The Opsono-Adherence Assay analyzes the presence of functional opsonic antibodies produced in animals after vaccination, which have been shown to correlate with the efficacy of several licensed human vaccines. This assay is preferred for use with biological select agents and other infectious pathogens and can be performed in a BSL-1 or BSL-2 lab after the inactivated BSL-3 agent is transferred.
Antibodies against bacterial capsules, including the Bacillus anthracis capsule, have been demonstrated to be efficacious in animal models of infection. Opsono-adherence assays functionally evaluate antibodies without infecting animals. Follow all institutional safety and security procedures when working with encapsulated virulent strains of Bacillus anthracis. For this video, we are using an inactivated strain under biosafety level 2 conditions.
Begin this procedure with preparation of raw 264.7 cells as described in the text protocol. To plate the cells for the Opsono-Adherence assay, replace the flask with fresh medium and use a cell scraper to gently scrape cells off the flask. Pipette up and down to break up the cell clumps for easier cell counting. Plate 14,000 cells per well in a 96 well 0.15